in paper one, the concepts that we always test are the concepts of summary writing. In O level, they were teaching you in English summary writing. And I tend to believe that by that time, we never used to think that these things are applicable. Or even sometimes we run short of skills of answering certain questions in biology because our listening skill, our summary writing skills are not good. Now, paper one requires you to have those skills of summary writing. You read a question, you brainstorm the answers in your head. Then at the end of the day, you have to note, you have to write down something which is very brief that is answering the question. That is the summary writing skills that we are talking about. Therefore, how best can we revise for biology paper one? The first thing is that whenever you're reading for biology, we practice the questions. You get your sample questions, like for instance, let me show my screen. Okay, so for instance, if you know that today you're going to revise biology, but you want to put special attention to paper one. So what you do, you gather your questions. You gather your questions of paper one. You said that now today, these are the questions that I'm going to use to read. Then when you read a question, like for instance, when you look at this question here on the screen, you identify the topic from which it is coming from. Then, even if you feel that you can attempt it yourself without opening the notes, it is not proper to try it out. You leave that one, you leave that responsibility to the examiner to task you, to test you whether you know something. Now, what you do for you, you get your notes that, oh, this question here must be coming from enzymes. Then you get your notes of enzymes, you sit down, then you now look at the question, you understand the question. Then after reading, understanding the question, then you see now how is this question answered. You open the notes. Are the notes going to be of help? Then if you find that yes, it is true, the notes are going to be of help, then what do? You now write or you now pick out your answer. Then you see how best you can summarize it so that the answer fits in the few spaces or in the few blank spaces that they have left for you to fill in. That is the first trick. When you do that one, remember, even as you're opening your notes, as you're opening the book, there are some things that are going to attract your attention. For instance, maybe you're looking for the role of how inhibitors work, the competitive and the non-competitive inhibitors. Then when you do that one, as you're opening the notes, looking for these competitive inhibitors and non-competitive inhibitors, your eyes are going to land on some things in your notes that perhaps you have taken a lot of time without revising. Then we'll say, oh, wait a minute. You first go through it briefly about a minute. Then you proceed looking out for what you want. Realize that by the time you answer about 3 or 4, the whole syllabus in less than an hour. So that is what you should always do. Read by practicing the question. Then, now, let us look at this question here. Now, today's seminar is more going to be teacher-centered. I'm going to take up the role of the facilitation because I know we don't have much time. So I'll just be going through these questions. Then I give you the techniques or the skills of how you can construct a quality biology answer. The answers have to be quality. They have to be short, brief, but at the same time of good quality. Now, when you open a paper and you go to section B, Number 41, you find a question like this one. Now, what you should always do, don't rush at looking at the kind of questions that you're going to answer. For example, these ones here. You don't rush to look there. If possible, you first cover the questions like that, because they will corrupt you. They will corrupt your thinking. You'll find when the questions are very difficult, then as you try to think of the answer, you want to go back and understand what the question is all about, then you can't have the liberty to think outside of the box. Now, when you look at a question like this one, the first thing is you read through and you put yourself in the hands or in the shoes of the setter or in the shoes of the researcher. You ask yourself, how was this data collected? If there are words, if there are new words, or if there are words that are attracting your attention, if possible, you underline those words. You say, oh, this one, like for example here, they are saying that the figure one below shows the relationship between substrate concentration and the initial rate of reaction of an enzyme catalyzed reaction under different conditions. Now, what do you do? First, when you look at the word initial rate of reaction, that word seems to be new. It's not a common word, because in most cases, the question that we find, they just say the rate of reaction. But now here, why have they said initial rate of the reaction? So what you do here, you just underline the word initial. So, okay, let me underline this word. I have noted it. Then now you go to the question. We have three graphs there. The first one, labeled A, we have no inhibitor. Then the second one, labeled B, there is a competitive inhibitor. Then the third one, labeled C, there is a non-competitive inhibitor. Now the question would be, when you look at the graph on the vertical axis here, we have initial rate of reaction. Then here we have the substrate concentration. We can see from the graph what we are seeing. You can see that as the substrate concentration increases, then what happens to the initial rate of reaction? The initial rate of reaction also increases. Then when you look at B, the same thing, C, the same thing. But however, we are able to note out some differences. We're able to note out some differences. Differences, like now, when you look at curve A, initially it is increasing rapidly. Then what of curve B? When you compare curve B with curve A, all of them are increasing. So this one is increasing rapid. This one for curve A. Then this one, we shall say this one is increasing gradually, but also this one is also increasing gradually. Look at this one here, the one for curve C. Now, what would be the difference? The difference would be that when you look at curve A, it increases rapidly with increase in substrate concentration and later attains a maximum at a high substrate concentration and remains constant. Then what of B? B increases gradually and attains a maximum at a much higher concentration of the substrate. I'm now identifying out the differences. So this one, curve A, attains a maximum at a high concentration of the substrate. Then for B, I'm saying it attains a maximum at a much higher concentration of the substrate. Then what of curve C? Curve C also initially, it increases gradually and later attains a maximum where it remains constant at a lower substrate concentration. So take note of the words that I have used. I have used words like high substrate concentration. I have used words like much higher substrate concentration. I have also used words like at a lower substrate concentration. So it means that if I'm discussing with someone, maybe on phone, like for instance, if I remove the screen and then I start describing to you the graph that I'm looking at, you should be able to sketch it. So that is the reason why we always ask you to describe. That can someone try to sketch the graph that you are describing by just reading what you have written? If they can't, then there is something missing in your description. So always take note of that. So that is one of the best ways that we can describe, or we can state, or we can compare those three graphs. Hope that one is clear. So it means I paid attention to the point at where the graph remains constant. Then also I wanted to point out that when you look at this graph here, the one for B, okay. So when you look at B at this point, beyond this concentration here, all of them, B and C, are increasing gradually. Now, but what is the difference? The difference is that curve C attains a maximum at a much lower concentration, whereas curve B attains a maximum at a much higher concentration. That is what I'm seeing from this thing here. Then, then also we now have to ask ourselves questions. Remember, we have not yet looked at the questions that we are going to answer, and never rush to go there. First, pay attention to the graph, interpret it, ask yourself some few questions, answer them so that you can be able to get a general knowledge of what this question is all about, or what the graph might be about. So A, where we don't have an inhibitor, is now going to serve as our control experiment. Because now this graph is showing you that if you let this setup take place without adding any inhibitor, you're going to get a curve which is in the shape of A. But however, if you add their inhibitor B, what is going to happen to your rate of reaction? The rate of reaction is going now to slightly decrease. Then what happens when you add competitor C? The rate of reaction is going to rapidly decrease. Why am I saying rapidly decrease and gradually decrease? Because I'm using A as my basis. I'm using A as my control experiment, as my basis. What happens to B in comparison to A that B reduces the rate of reaction? Because normally the rate of reaction was supposed to be this one of curve A. But now what has happened here, it has reduced like that. Let me first finish this thing, then I will allow you time for questions. Then also another thing that we can point out here is the use of the word initial. Why use the word initial? Why they say initial rate of reaction? Why shouldn't they just say the rate of reaction? So you have also to ask yourself, what is the meaning of this word initial rate of reaction? And by the way, rate of reaction is different from initial rate of reaction. They are different. Rate of reaction happens like this. You get your substrate and you also get your enzyme. You bring them together. Then they start reacting. Then as they start reacting, you see the intensity of the reaction over time, over a given period of time. So that is the rate of reaction over a given period of time. Then what of the initial? the initial rate of reaction. For you, you're just interested in the rate of reaction at the start of the experiment. That is the initial, at the start of the experiment. What happens afterwards, that is none of your business. So it means when they were determining, when they were getting this data here, when they get a concentration, a substrate, when they get substrates of about maybe 5 molars of concentration. Now, they react this concentration, this substrate at 5, whatever percentage of concentration you have, whatever units of concentration you have used, then they react it with the enzyme, and they only capture the rate of reaction at the start of the experiment, and they get that figure. They note it down. Then they increase the concentration to around maybe 10. Then at 10, they also add in the enzyme. Then the reaction starts. Now they only look at the rate of the reaction at the start of the experiment. What happens later, we don't record that data. So they keep on doing that one until the whole graph is covered. So why should they use initial rate of reaction? Now remember, when an enzyme reacts with a substrate, definitely products are formed. So if we have a reaction where we have a large amount of products and also a large amount of substrates, now that one is going to reduce the rate of reaction. How? Instead of the enzyme colliding with the substrate, it will have also to collide with the proteins, with the products. Then after colliding with the products, then it realizes that it has hit a dead end. Then it detaches, goes and looks for substrate. All that is time wasted and reducing the rate of the reaction. Therefore, it means that it is important to always take the initial rate of reaction, because the accumulation of the products have a significant effect on the rate of collision of the enzyme with the substrate molecules, whereby the enzymes, instead of colliding with substrates, they'll also collide with the products, hence minimizing the rate of the reaction. So if you want to get very accurate results, then you consider the initial rate of reaction, like that. So I will entertain some few questions, then we proceed. Carol is complaining that she's not hearing at all. Rebecca is saying, which mock are we discussing? We're not discussing any mock exam here. We are just discussing questions in paper 1. Dan is saying, teacher, can I record? Danny Cruz, I think, let me see if I can give you the recording rates. Let me first see if it is possible. Okay, yes, Danny, I've given you rates. You can record on your computer. Then Hassan is saying, Master, do we have descriptions with the trend or without trend? Okay, I will talk about that one. Then Someone is saying, teacher, look at UNEB 2013. Okay, we shall also discuss that question and we see. Richard is saying, why is C initially increasing gradually? Why is initially increasing? Okay, we shall discuss those things. Then William, what does it mean by graph has attained the maximum and a peak? Okay, now William, a graph, we always say a graph has attained the maximum or a peak if these things happen. If the graph increases and then reduces, then we say it has attained a peak. If a graph increases and remains there constant, then that is a maximum. So if it remains constant, we say the graph increases to a maximum and remains constant. Then if it increases and then decreases, that is a peak. We say it has increased to attain a peak, after which it must decrease. Then when is initially used when describing? Now, when do we use initially? When it's describing. If we don't have units or if we don't have figures on the horizontal axis, then we can use the word initially. Now, for example, if we are to describe this graph, would we use the word initially? It would be inevitable. It would be unavoidable because we don't have figures on the horizontal axis. So we use that word when we don't have figures on the horizontal axis. That's when you can use words like initially, later, afterwards, etc. Someone is asking, sorry about that. Someone is asking, it's similar, they are asking the effect of the inhibitor on rate of the reaction. We shall talk about that one, clarifying whether to state trend and then qualifying. Now you're going in very complicated words. But we shall talk about that one when I'm answering that question. Teacher, help me and show me the maximum for curve B. Okay, the maximum for curve B, it is here. This point here, that is the maximum for curve B. That point there, that is the maximum for curve B. Then explain the difference in the effect of inhibitor A would be equal to A when except we tabulate for comparisons on graphical numbers. Now, when you're giving differences, you do what you want. You can either tabulate the differences or you may not tabulate them when you're doing graphical questions. So you do what is convenient. There are no restrictions that when you're doing graph numbers, you make differences. You do what is convenient. Then how? Okay. Then, so let us now first go to the question. Okay. Then now we'll accept some few hands. Yes, Muhindo. Muhindo, what's your question? Okay, Muhindo is not there. Okay. Now, let us now go to the graph, to the question now. So now, after understanding what the question is about, then now let us now look at the questions. Question number 1, suggest why the initial rate of reaction was measured in each case. So the something which I had just brainstormed, which I've just explained to you. Now the question is there. Why is that? It is the initial rate of reaction that was measured in each case. Yes, Mohindo. Mohindo, can you ask your question?
I've just joined, so I wanted you to explain.
Well, if you have joined late, please, it is your fault that you're late. It is not our fault. Just try to start following from where you joined. Nega, you have a question? Ruth? Is it my network or Mike?
Okay. Hello. Question is, how you begin the graph at what
Mike, we can't hear you. Your network is not good. Let us now proceed. So now here, I had already described it to you, or had already explained to you why we should consider the initial rate of reaction. Simply, you know that when we don't use the initial rate of reaction, accumulation of products always have a significant effect on the rate of the reaction. Therefore, we take the initial rate of reaction before products accumulate, like that. Okay. Then, the network of some people. So that is it. Then now we can go to Roman number 2. Explain the shape of curve A. There is someone whom I'm failing to mute. The network is slow. But however, if you have unmuted yourself, kindly mute yourself. Okay. Now, they want us to explain the shape of curve A. Now here, when they ask you to explain, it means that they are saying the curve, the shape of the curve. So you look at the shape of the curve, you develop an explanation for it. And you can't explain something that you have not described. You first describe and then explain. Even in the paper 1, though your description should always be very short and brief, because you don't have enough space to rate whatever you want. I told you paper 1 tests for this summary writing skills. Now, they want us to explain the shape of curve A. Even look at the marks, only 2 marks. Now, let us go to curve and see, and they show you how it can be explained. So they want us to explain the shape of curve A. First of all, you have first identify out what is its shape. If you're describing someone, the shape of that graph on a phone, how would you describe it? I would simply say, initially, the graph increases rapidly, and then later remains constant. Full stop. I have described the shape of the graph. Let me repeat. You say that initially, the graph increases rapidly, and then later remains or becomes constant. That is the shape of the graph. Now, what if they ask me to describe if they ask me to describe the initial rate of reaction and the substrate concentration for graph A, or to describe the effect of the substrate concentration on the initial rate of reaction for curve A. Now there, I should even mention the word substrate concentration. I should even mention the word initial rate of reaction. For instance, I will say that as the substrate concentration increases, the initial rate of reaction rapidly increases and then attains a maximum point at high substrate concentration. That is it. So if they ask me to describe the shape, I'm just interested in the shape of the graph. And what is the shape? Increases rapidly and then remains constant, full stop. Then now, can we develop an explanation for that? Because the question is saying, explain. So after writing my description, I say that the shape of the curve increases rapidly initially, or initially increases rapidly. Why? Now I go back to my biology. Why? Because increase in substrate concentration increases the chances of the enzyme molecules colliding with the substrates. Hence, they observe the rapid increase. Then full stop. Then I again say, at high substrate concentration, the initial rate of reaction remains constant, or maybe the graph remains constant. Why? Because all the active sites of the enzyme are occupied. Full stop. That's all. That's what that question wants. You don't talk about there's so much biology. Come on, this is paper 1. And more so, we only have 2 marks. Like that. So let me repeat briefly the answer I've told you. I've told you that the rate of reaction, or maybe here for Roman number 1, I've told you that the graph initially of increasing substrate concentration. Like that. So I've told you that here I said that the curve or the graph increases rapidly initially. Why? Because of increase in substrate concentration. Then the graph remains constant at high substrate concentration. Why? Because all the enzyme active sites are occupied, just like that. You just keep your answer short and brief. You're working for 2 marks. They are not much, but they can contribute a lot. So you still don't have to ignore them. Then the next question, explain the difference between the rate of reaction of curve B and curve C. Okay. Now, here, they are saying they want you to explain the difference between the rate of reaction of curve B and curve C. Now, the first thing to do is to pinpoint, is to pick out what is this difference that they want me to explain. That should be your first question. What is this difference that they want me to explain? Therefore, I have to pick out that difference. I say, this is the difference. Then I give an explanation for it. So that should be the first thing. Identify that difference, pick it out, write it down, and then explain it. So what is this difference that we are seeing? Let me go back to the graph and we see. So they want us to explain the difference between curve B and curve C. That's what they want us to explain. So what is the difference that we are seeing here? I want to first hear from you. What is that difference that they are asking for? Jerome, I just want someone to just describe for us the difference, or to tell us the difference between curve B and curve C that we should explain. Yes, Jerome.
Hi, teacher.
How are you?
I'm good. Yes, teacher, according to me, I'm saying that generally the rate of reaction, or according to curve B, is higher than that of curve C.
So curve B has a higher rate of reaction than curve C. That is correct. Another alternative? That is correct. Augusty, Mike.
Thank you. According to me, I am looking at the curve B attaining its maximum at a higher concentration compared to curve C. You realize that curve C is attaining its maximum peak at a lower substrate concentration compared to curve B. Thank you.
Yeah. Thank you. So you're also correct. So for you now, you have paid attention to the concentration of the substrate where they attain the maximum. Then let me have a lady. Do we have a lady now? Jane.
She's over participating. Hello.
Jane, you can unmute.
Teacher, I think curve B increases gradually to a higher maximum. Then graph C increases gradually to a lower maximum.
but now putting it into proper words, now the challenge. Now here, I think what you should have said, you could maybe say that curve B attains the maximum at a much higher concentration, and curve C attains a maximum at a much lower substrate concentration. Or maybe you can even say that curve B attains the maximum, it attains a much higher initial rate of reaction. You say curve B attains a much higher maximum rate of reaction. Then curve C attains a lower maximum rate of reaction. That here the maximum, the initial rate of reaction at maximum is lower, then this one is higher. Okay. Then Agupat, Angu Patrick, I don't know what it is.
Teacher, what if I say that curve B attains, curve C attains its maximum faster than curve B? Am I right?
Faster. Now, when you say faster, it means that for you on your horizontal axis, you have time.
Okay.
Yet it is substrate concentration. If this one was time, then it would be correct. This one faster, then this one later.
Okay, thank you, teacher.
You're welcome. Someone using ITEL?
Okay. Thank you, sir.
So
okay,
fast.
My rate of reaction whereby curve B initially reacts more rapidly than at a low rate of at a low substrate concentration. Curve B reacts more rapidly than the reaction of curve C.
You also have very good idea, correct, but now getting the correct words to put your ideas on paper is also now the challenge. But though you know what should be done, but now getting the right words to use tends to be the challenge. Okay, now here, it doesn't matter how you give us the difference. So me, I'm also going to give you mine. And by the way, none of you was wrong. None of you was wrong. All of you were correct. You'll get the marks. Now we'll just be waiting for the explanation, where also the explanation is correct. But you'll at least get the first marking point. Now, me, the first difference that I would point out would be that curve A increases and attains a maximum at a much higher concentration of the substrate. Whereas curve B also increases gradually at first, but attains a maximum at a much lower concentration of the substrate. Because, then now we start now thinking of the biology behind that. And when you are comparing, because here there is explain the difference, first state the difference and complete it. Then after completing the difference, then you now give the explanation. Don't say that curve B increases gradually and attains a maximum at higher substrate concentration because of this and this, full stop. Curve C does this and this because you have not given us the difference. You have first give us the difference. Okay, so what then would be the explanation? Remember, I've told you my description, my difference, that curve B increases gradually and attains a maximum at a much higher substrate concentration, whereas curve C initially increases gradually and attains the maximum at a relatively lower substrate concentration because, then now I start brainstorming the biology behind it. Why? So, of course, they have told you B is a competitive inhibitor and C is a non-competitive inhibitor. So if it is like that, then what? So it means you can now explain to us how B would result into the initial rate of reaction, the maximum initial rate of reaction being attained at a much higher substrate concentration. And why is it that the non-competitive inhibitor for it attains the maximum at a much lower substrate concentration? That is what we should now be asking ourselves. Now, to keep my answer brief, I would simply say that, now I'm on the because part of it. I would say because in curve B, the competitive inhibitor competes with the substrate for the active sites. Therefore, an increase in substrate concentration increases the chances of the substrate colliding with the enzyme instead of the inhibitor. That's it. If it again sounds long, then you also have again to compress it until you get a statement which is very brief, but at the same time, accurate. Then for curve C, I would say the non-competitive inhibitor in curve C attaches on a site other than the active site and completely distorting the shape of the active site. Therefore, very few enzymes that are not inhibited will still proceed with the rate of reaction, will still proceed with the reaction. That's it. Now, what does it mean? It means that here, when here the rate of reaction is increasing, of course, it's increasing gradually. Why are we saying gradually? When you compare it with the control experiment, the control you can see it was very rapid. But when you applied the C, the rate decreased compared to the control experiment, that is curve A. Why? It means that now here, the enzymes were still being, the substrate were still being attached onto the enzymes. Then when we reach here, most of the enzymes are inhibited permanently, leaving only few enzymes to proceed with the reaction. It doesn't mean that all the enzymes are inhibited. No, because who knows, maybe the number of the competitive inhibitors are also few. So if you want to further decrease this rate of reaction, then you introduce in more of the inhibitor, like that. Now, there is a question which they can ask you, the one that appeared in 2013. 2013, let me first see if I can open it quickly here. Okay, so let me first see if I have that here very fast. 2013. I used to have the question banks here. Let me look for it. Yeah, I think I have seen it. So this is 2013. Here is the question. So now here, it is similar to what we have been discussing, not so different. Push it down. It is not so different. So here, the first graph is for the control. That is the inhibitor. Then for this one, we have inhibitor A, and then we have inhibitor B. Now, if you compare it with the question that we've just from discussing, for them, they called this one the non-competitive. Then this one was now the competitive. So therefore, how can you answer such a question? Now, when you look at the questions asked here, of course, they are saying that describe the effect of each inhibitor on the rate of reaction. And they're starting with inhibitor A. So they're asking for the effect of inhibitor A on the rate of reaction. Now, here, I know, though, because we're running out of time, I will not first, now, I know if I had asked you to give me the answers here, many of you would have said that the rate of reaction increases with substrate concentration. You'd be switching to say that this one, there is a gradual increase, then it remains constant, like that. But what is the question saying? The question is saying the effect of the inhibitor A on the overall rate of reaction, and not the effect of substrate concentration, because that is another question. They can ask you to describe the effect of substrate concentration on inhibitor, on the reaction in presence of inhibitor A. That question is totally different from this one here. So it's like they're asking you that when you look at this graph here, what happens when we add inhibitor A to the rate of reaction? What is its effect? In graphs where they have given you control experiments, all your descriptions should be based on control experiments, on the control experiment. So here, what happens? You can see when you add the inhibitor, the rate of reaction decreases. Why are we saying that it decreases? Because normally it is supposed to be here. Normally it was supposed to be here. Just a moment. Let me put this thing down. Okay, there. Now, this graph was supposed to be here. The rate of reaction was supposed to be at this level here. But what happens when we add the inhibitor? It reduces from this level to this level. Then what happens when we add inhibitor B? Look at it, it reduces from this up to this. So it means this is a gradual decrease, and this is a rapid decrease. That is how the inhibitors are affecting this reaction here. That inhibitor A inhibitor A gradually reduces the rate of reaction compared to the control experiment. Then inhibitor B rapidly or greatly reduces the rate of reaction compared to the control experiment. There I have described. That is what that question is asking for. What is the effect of this inhibitor? Not the effect of substrate concentration, no. The effect of the inhibitor. Hope that one is clear. Okay. So let me now go back to this question here. So Roman number 2 we have finished. Then now, before we go to figure 2, let me look at some of the questions that we may be having here. Let me check in the chat room. Ivan is saying control experiment according to this question means that. It means that or means what? Then what is a control experiment now? A control experiment is that experiment that you do when certain parameters are controlled. You want to know, you want to see if the inhibitors have an effect on the rate of reaction. That is what this person wanted to know here. So in this case, our A is the control experiment. So the person wanted to know, what is the effect of inhibitor B on the rate of reaction? So what you do, you set up another experiment where you don't have inhibitor B, and another experiment where you have inhibitor A. So that will be able to clearly identify that it means inhibitor B does this. Why? Because where it isn't, I have gotten other results. So that is the control experiment, where you control certain parameters in the experiment or in the setup. Or sometimes you can even say it can serve as the basis. The control experiment is the basis of your experiment, like the reference point of your experiment. Okay, let me check again in the chat room. Xavier is saying, what if you consider the curve C getting a constant, yet B doesn't have? Please, on that question, curve B also has a constant. It is there. Pardon, an explanation for curve B. Help me with your phone number. Okay. I've just put my phone number in the chat. You'll find it there because when people are asking for it. Okay. Now, let me also pick one question and see. Competitive inhibitor. Okay. So now, if they ask you, the explanation for how inhibitors work are non-competitive. A competitive inhibitor competes with the substrate for the active site. They compete. But a non-competitive inhibitor does not compete. For it, it binds at a point other than the active site, and ends up changing the shape of the active site. So that is the body that you should go and look at in your notes, because those things must be there in the notes. Because now for me, my role here is to help you, show you how certain questions are answered. That's why I don't give you a direct answer, I'm just giving you a hint. Then you can go understand the biology from your notes, then know how to apply that biology in answering certain questions. Okay. Then now let us look at this question here. So here is the question. So they are saying figure 2 represents a metabolic pathway controlled by enzymes E1, E2, E3, and E4. Then they're asking you, name the type of control mechanism. So, what is the type of control mechanism this is? This is just, it is called allosteric inhibition. Or you can simply call it end product inhibition. That is the resolution, the control mechanism here that is controlling the production of compound Z. Then they're saying, explain how an excess of Z, will inhibit its further production. Now here, you just look at the graph, the figure, and you ask yourself, how does excess of Z inhibit production of Z? Even you can see clearly here, there is a line showing you that product Z will come and inhibit enzyme E1. Therefore, what should you say? You just say high concentration or excess of compound Z inhibits enzyme E1. Hence, products W, X, and Y will not be formed. Just like that. Others might have thought that maybe the answer is supposed to be very complicated. My battery is warning. Just a moment, let me pick the charger. Okay. Okay. So the answer I have told you that, because we're answering here that explain how an excess of compound Z will inhibit its further production. From here, what you can see here, product Z will inhibit enzyme E1, is controlling or inhibiting the production of these subsequent products. That's it. You don't now have to go into the details of that complicated biology. You don't have to go into those details, because here they just wanted to focus on compound Z. But however, we know that this concept here of end product inhibition, they can as well ask you, they can ask you to explain how allosteric inhibitors work. They can ask you that question. So how do allosteric inhibitors work? Allosteric inhibitors, these are the inhibitors that have a binding site on an enzyme. There are some enzymes that have special sites only reserved for allosteric inhibitors, not for the substrate, but they are always reserved for the allosteric inhibitors. Any other compound will not fit in. It is only reserved for the allosteric inhibitor. Therefore, if they ask to explain how do these inhibitors work, how does allosteric inhibition work, you just simply say that in a reaction which takes place in a series, accumulation of the end product or the end product, an excess of the end product inhibits the enzyme controlling the first step of that reaction. Just like that. That first of all, you have to acknowledge that this allosteric inhibition works in processes that take place in a series. Therefore, the end product will always inhibit the enzyme controlling the first step. So when you look at this question here, this end product Z does not inhibit enzyme 4, does not inhibit enzyme 3, does not inhibit enzyme 2, but it only inhibits the enzyme 1, which controls the first step. That is how allosteric inhibitors work. Okay, so that marks the end of that question. So I hope we've gotten some skills of how certain questions can be answered. Someone is asking how do you know it's an end product inhibition? The figure itself, is telling you that there is an end product here and it is inhibiting the first step of the reaction. So it is clear. Then someone is asking how non-competitive inhibitors and allosteric inhibitors differ. That is a very good question. Now, what is the difference between a non-competitive inhibitor and an allosteric inhibitor? Now, all enzymes can be inhibited by non-competitive inhibitors, all enzymes. But not all enzymes can be inhibited allosterically. Why? We have what we call allosteric enzymes. Now, allosteric enzymes, for them, they have a special active site or a special binding site reserved for only the allosteric inhibitor. So it means that now allosteric enzymes or enzymes that can be inhibited allosterically, they have two sites. They have the active site for the substrate, and they also have the active site for the inhibitor. Those are now, that is the allosteric inhibition. Whereas the non-competitive, for it, it doesn't have a binding site reserved for it. For it will just come and attach anywhere on the enzyme. After touching anywhere on the enzyme, then it ends up distorting the shape of the active site. So that's the difference between allosteric inhibitors and non-competitive inhibitors. That allosteric inhibitors, they have their special sites reserved for them on such enzymes that they inhibit. So they don't inhibit each and every enzyme. Now, when you look at this question here, it means that E1 doesn't have a site where the end product Z can bind. E3 also doesn't have a site where the end product Z can bind and inhibit allosterically. But it is only enzyme E1 that has that site like that. So I hope that one is now clear. Then someone is saying, what happens when I remove E1? The reaction stops. Aha. Ivan is saying, should we say that allosteric enzymes are specific to only inhibiting enzymes catalyzing the first step in the experiment? Yes. They only specifically bind to enzymes controlling first steps of a reaction, but for only those reactions that can be inhibited allosterically. So for them, they just for the first enzyme. What if I say chain reaction? Yes, chain reaction is also all right. Is also right, but now the most correct word is a reaction occurring in series, not chain reaction. Then Trina. Trina, yes, Trina was trying now to answer Ivan that allosteric enzyme is an enzyme that has an additional binding site. That is true. It has an additional binding site for the inhibitor molecule. Then Hassan is saying types of inhibition. That one you'll find out in the notes. Okay, so I think those are the questions, the serious ones. Okay, so we can now go to the next question. Now, the next question is coming from histology. I don't know, those ones in senior 5. I don't know when you last revised histology. Or I don't know when you last found a question coming from this topic of histology. Now, when you find a question like that one, now what should be your role? You go, get the question, and also look for your notes. Then when you get the notes, you read through. Then you get out the answer and you transfer the answer. So now here, they're saying state 2 locations in the body of a mammal and the importance of the tissues in these locations. First of all, you have to interpret the question well. This question is asking for 2 things. One, they want the location. Then 2, they want the importance of the tissue in that particular location. Then I have to think about your presentation. How are you going to present your answer? What you do here for the squamous, it means you'll give us the first location and the importance in that location. You give us the second location and also the importance in that second location. You don't mix up things. Some of you have a tendency of mixing up things. They ask you for differences and they ask you for a comparison. So you have maybe differences and similarities. For you, just join everything. First point is a similarity, second point is a difference, and you want the examiner to help identify that, oh, here she was meaning a similarity. Oh, here she was meaning a difference. No. Then, so now here, it means that for every location that you give us, also give us its importance, like that. Now, if I may ask you, I want you to give me one location, and maybe let us first brainstorm on the locations of the squamous. Where do we find the squamous epithelial tissues? You can now write in the chat. Where do we find the squamous epithelial tissues. Where do we find them? The locations. Okay. Alveoli, Bowman's capsule, lung alveoli, blood capillaries, lining walls of alveoli, Bowman's capsule, skin. We don't have squamous in the skin, please. Buccal cavity of the mouth. We also don't have squamous there. In the buccal cavity of the mouth, we have there stratified epithelium. They are not squamous. Sometimes they call them stratified squamous epithelial tissues. Endothelium, yes. Walls of alveoli, yes. No, we don't have oviducts. Sorry, we don't have epithelial, squamous epithelial in the oviducts. Then when you say ileum, where exactly in the ileum? It must be the lining of the villi. Joseph, heart chambers, no, we don't find them there. Renal capsules of the kidney, no, we don't find them in the renal capsules of the kidney. You have to be specific. Lymph vessels, lining of the gut. Lining of the gut, I've said no. Lining of the vagina, no, we don't find it there, the squamous. Okay, so I think that's enough. Now, the squamous, these ones, they are characterized by one, they are one cell thick, the squamous. They are one cell thick. The cells we find lining our mouth, the inner lining of the mouth, they are not one cell thick. Because even when you get a toothpick, or maybe you get something and you rub, you remove some of the cells. Maybe you can get maybe the cotton bud. When you rub in your mouth, even when you're eating, some of the tissues get worn off from your mouth. And others come. Others are replaced. They will be replaced. Now, those are not the squamous epithelium. Squamous epithelium, they're just one cell thick. One cell thick. And they have a smooth surface. They don't wear off. They don't tear off. Remember the ones you find in your mouth, those ones, they tear off. And when you rub something into your mouth, you remove very many cells from that tissue. So take note of that. Now, where do we find them? We can find them in the endothelium, or the inner lining of the blood vessels. And not even the capillaries. By the way, capillaries don't have squamous. So we find them in the inner lining of the blood vessels, including the capillaries. Inner lining, not the entire capillary, but the inner lining. And what are they doing there? They provide a smooth flow of blood, because their surface is very smooth of these epithelial. So we find them insects where they can provide a very smooth flow, so that, because if they were very rough, then blood will be not flowing smoothly through those blood vessels. So that is location number one, and the importance. For every location that you state, you also state its importance immediately. Don't first outline the locations, then after you give the importances. Then you ask yourselves, now, this importance is for what? Then we can also find these squamous lining, in the alveoli. The alveoli walls are made up of squamous epithelium. And what are they doing there? Gaseous exchange. They're doing the role of gaseous exchange. Okay. I was trying to check whether there are some questions in there. So that's what we need there. Now, I want to give you a task of reading, revising. When you open your histology notes, look at all the tissues that the teacher has taught you. And from each and every tissue, this is what you need to know. One, you need to know on each and every tissue under histology. You need to know the structure of that tissue, all the characteristics of that tissue. You need to know the location of that tissue, the importance of that tissue, and how it is adapted for, how it is adapted to its function. Those are the things that I want you to revise for each and every tissue, including the plant tissues. Plant tissues like parenchyma tissues, collenchyma tissues, sclerenchyma tissues, xylem tissues, phloem tissues, all of them. Ask yourself, or when you're reading, look out for the structure or the characteristics. Look out for the location, be it in a plant or in an animal. Read about the functions of these tissues and also how they are adapted to their functions. For instance, we have seen the squamous. The characteristic of the squamous, we've said, they are one cell thick, they are very thin, and their surface is very smooth. So how are they adapted to their functions? In the lining of the blood vessels, they have a smooth surface, which provide a smooth flow of blood. That is an adaptation. What of the one we find in the alveoli? They are one cell thick to reduce on the diffusion distance during gaseous exchange, or to speed up gaseous exchange by reducing on the diffusion distance. Just like that. Okay. Someone is asking, what is their role in gaseous exchange? They provide a very thin lining which reduces the distance over which gases they fuse. Then Joel Musingu's formation of plant vascular tissues. I've not got that one. I don't know what the question is. I've not got your question well. Okay. So now we can proceed. So I've guided you how you're going to revise the whole of histology and finish it. And when you do, then you can go and attempt this. I'm going to share this question immediately I finish this session. I'm going to share this paper here. You try it out. Now, some of you, sometimes you send me questions, answers on the WhatsApp, on my WhatsApp number. Yes, sometimes if I have the time, I can go through. But if I don't have the time, I may not even open to go through. So it will depend on the time that I have. Because now these days I'm too occupied. So if you like and if I have the time, I will go through your work. But if I don't, you know, you can read an answer and you know whether it is correct or wrong. Because remember these things, I'm not telling you that you go and you test whether you have these things in your head. Read the question, open your notes, look out for the correct answer. In that, even when you send, don't say, maybe teacher, mark me. Don't you know that what you have written is right or wrong? You already know because you used the books. And that's what I'm telling you to do, use the books. So maybe if the question, if you're not so sure with the approach, or maybe if you're not so sure with the interpretation of the question, then that's when the teacher can come in to guide you. Otherwise, don't think that even when you read the definition of osmosis, then you want to send that on the teacher to mark you. To mark you what? Then if it is correct, if it is wrong, what have you gained? So when you're reading these things, please use notes or textbooks. Extract the correct answer. Read through to make sure that what you have written is actually correct. Then after that, you'll have revised. Okay, so I want now to go to part B. And remember, we are not here to cram the answers. We're using these questions to help us as case studies, to help us know how can an answer be developed. How can an answer be developed? Okay, so we have part B. State two structural differences between the epidermis of the mammal and the epidermis of the plant leaf. So they now want the differences between the epidermis of the mammal and the epidermis of the plant leaf. What are the differences? Now here, still, some of the questions that they ask you, you don't always have to sit down and say, but now the teacher didn't teach us these things. Now where's the answer? You just look at the things and you get the specimen and you copy from them. Now here, what is the epidermis of a mammal? That is your skin. Then what is the epidermis of a leaf? We've been drawing structures, internal structures of a leaf. You know that a leaf has the internal structure and also has the upper epidermis and the lower epidermis. Now they're asking you now, what is the difference between that epidermis of the leaf and also your skin? Because your skin is the epidermis. So what are the differences? You just read from, you look outside, get the leaf, and you look at your skin. Which differences are you seeing? My skin or the epidermis of a mammal has sweat pores. Then what of the leaf? The epidermis of the leaf has stomata pores. That is the difference. When you look at the one of the leaf, it has a cuticle, a waxy cuticle. Yours doesn't have a waxy cuticle. Then what of, look at the hairs. The epidermis of a mammal, or maybe your skin, has hairs embedded within the epidermis. What of the leaf? The leaf may or may not have hairs on the epidermis. Those are the things, the differences. And remember, the best way to rate a difference, you can draw a table. But even if you don't draw a table, they won't penalize you. But it's always much easier when you are drawing, when you're setting differences in a table format. Okay. So that's all with that number 2. Okay. Then we can now go to, I think, let me go to number 44. Now, maybe to briefly talk about this one here. This number 43. Now, number 43 is a question that comes from field work. I don't know if you've gotten a chance to be taken for a field trip, a biological field trip, the study trip for biology. And we always tend to notice that pollution is always more abundant in urban areas. Why? Because there are very many industries in urban areas. Now here the question is saying the number of ocean species growing along a 20 kilometers transect from the city center was recorded at 1 kilometer intervals. The results are presented. So what do we see? Distance from city center, and then here, number of lichens. So what are we seeing here? We can see that as we move away from the city center, the number of lichen species increase. The number of lichen species increase. Now, they ask you, look at the questions. What is the relationship between the number of lichen species and the distance from city center? What is the relationship? Now, this question is not asking you to describe. You just look at it and you look at the relationship. You connect the relationship between number of lichen species and the distance from city center. Just then you give us that when I look at this, this happens when this is happening. That's what they mean by the relationship. Otherwise, you don't have to go into think that from 0 to 5, number of lichen species increase gradually. From 5, for maybe around 10 to 15, number of species increase rapidly. There, you are describing. You are not telling us the relationship. I hope that one is clear. Now, what would be the relationship here? What do you see? As distance from city center increases, number of lichen species also increase, except at the 10-kilometer spot where number of lichen species decrease. I've just given the relationship. Number of lichen species increase as distance from city center increases. And what can be the reasoning? Hey, the reasoning, pollution and its effect on lichens. Lichens are indicator species. Indicator species, these are the species or organisms that can show the quality of the environment in terms of pollution. There are some species where the environment is highly polluted, you'll not find them there. But if you find them in an area, then it will be an indicator that actually there is no pollution here. Those are what we call indicator species. And lichens are part of the indicator species. So now, what would be the explanation here? It means that as you move away from the city center, the level of pollution decreases. And once the level of pollution decreases, then what happens? The number of lichen species also increase. So that is what it means. That as we move from city center, the levels of pollution decrease, which results into an increase in the number of lichen species. Because we find lichen species in regions that are not polluted. And what exactly in the environment destroys these lichen species? It is the accumulation of sulfur dioxide. So when sulfur dioxide accumulates in the environment, these lichens are very, very sensitive to these lichen species. Very, very sensitive. So their numbers will always decrease. So that is the reasoning there. Even the second question is asking you, let us see. Explain the relationship described above. In other words, now they want you to explain why is it that as we move from the city center, the levels of lichen, the numbers of lichens increase. So I have told you, I've given you the explanation that as we move away from the city center, the levels of pollution decrease. Therefore, the amount of sulfur dioxide also decreases. Lichens are sensitive to amount of sulfur dioxide in the atmosphere. So if it decreases, then the number of lichens are going to increase. But in the city center, where we have the largest, where we have a lot of industries, a lot of pollution, a lot of sulfur dioxide. Now this sulfur dioxide is now going to kill the lichens, hence they are observed low numbers. Then what can be the possible explanation? Why is it that at a certain point, at 10 kilometers here, the number of lichen species reduce? So it's like all of a sudden you're moving away from the city center. Then you also find that a place, maybe like a trading center, where the number of lichen species have also declined. Now, what could be the explanation here? Maybe there is a dumping site there for garbage from the city center, which is polluting the area nearby. Maybe there is an isolated factory at that center, which is releasing gases into the atmosphere. Or maybe there is a small urban center there, which is also doing some of the pollution here. Okay. Someone is saying that color of the skin and leaf is not biological. Of course, remember the question was saying structure. Color is not a structural feature. Someone is saying competition between lichens. No, lichens do not compete. Now they are competing with who? Remember, lichens, for them, what they need, the algae in the lichens just needs sunlight. Then the fungi in the lichens, they need moisture. Once they get those things, they are good to go. So whom are they competing for? So there is nothing like competition here. The thing is just simple. Lichens work as indicator species. So whatever is affecting their numbers is related to the levels of pollution. Okay. Rodin is asking for a pardon as why number of species, number of lichens increase with the distance from the city. I've said that in the city center, we have a lot of industries. Even in the villages, you won't find a factory in the village. Factories are almost in the city center. Now, because we have a lot of industries in the city center, that also means that there is a lot of pollution in the city center. So as we move away from the city center, the levels of pollution keep on reducing. And once the levels of pollution decrease or reduce, then the number of lichens are going to shoot up. Just like that. Okay. Why is it that not all lichens decrease to zero, but slightly decrease, yet they are greatly affected by sulfur dioxide? Now, Ivan, it depends where. Because here you see, it at spot 0, the center of the city, we have no lichens. The number is 0. Why? A lot of pollution here, a lot of sulfur dioxide gas here. Then at this point, the levels of lichens reduce. It means that there is some sort of pollution here, though it is not much to reduce the numbers back to 0. So this one, and I've given you the reason why this decrease. Maybe there is an isolated factory at distance of 10 kilometers from the city center. Maybe it is an urban center, a small urban center from the city center, or maybe there is a dumping site for rubbish from the city center that is causing mildew. Okay. Which species of lichens will be more resistant to sulfur dioxide? You don't need those details. You don't need, at your level, you don't need the different species of lichens. Other examples of indicator species. We can have the toads in water bodies. If you find that there are no, for instance, if you find that there are no tadpoles in water, then that could be an indication that that water is polluted. Then we can also use mayflies. We normally use mayflies in testing the quality of the water. Mayflies, these are the flies that you always find. Sometimes they call them lake flies. These are flies you always find hovering over water. So those flies, they always like quality water. If the water is polluted, they will not be there. So those are also other examples of indicator species. Then what is the exact effect of sulfur dioxide to lichens? And now the effect of sulfur dioxide to lichens is that sulfur dioxide, once it is released in the atmosphere, it will react with rainwater. Then it will react with rainwater to form acidic water. This acidic water will come and fall on this lichen rain that eventually kills these lichens. And by the acid rain also has an effect on other plants, not only the lichens. Okay, so I'm going now to stop on this number. I'm going to stop on this number. Then when we finish this number, we call it a day. Now, I want you to write down this question and you briefly try it out. I'm giving you 4 minutes. Write down this question and try it out. 4 minutes. So write that question in less than a minute, then you attempt it. In that when I'm explaining, you're following. Okay. So I'm seeing some questions in the chat. Someone is asking an epistatic gene. Now, an epistatic gene. Now, an epistatic gene, there are some situations or there are some genes that for them to be expressed phenotypically, there must be another gene present. That their existence or their expression phenotypically depends on either absence or presence of another gene. Those are the epistatic genes. For instance, if maybe, let me just use maybe a big example. You may find that someone who maybe has, though it is not true, someone maybe can be having blue eyes. You may find that if someone has the gene for blue eyes without the gene for baldness, then that person will not develop blue eyes. But you find that only a person will get blue eyes only and only if he also has a gene for baldness. Kind of that scenario. That for a gene to express itself phenotypically, it depends on absence or presence of another gene. That is an epistatic gene. Then what about chlorosis of green plants? Now, chlorosis means loss of the green coloring pigment, means loss of chlorophyll in a leaf. Some leaves can start to lose their chlorophyll. Like for instance, if you put a plant in the darkness and for about 2 or 3 days, when you get out that plant, it will be appearing pale yellow. What has happened to it? It has lost chlorophyll pigments. So that loss of chlorophyll pigments is what they call chlorosis. Someone is asking that, do we have any other seminars after today's seminars? It will depend on the resources available. So you just keep your fingers crossed. If we happen to organize another one, you'll get to know. And for us, we shall be there to guide you. Someone is asking explanation on wilting. Now, wilting is when the plant cells lose water. Because we have wilting and plasmolysis. Now, under wilting, the plant cell loses water. But also, under plasmolysis, the plant cell also loses water. But for plasmolysis, the plant cell loses water by osmosis. Then wilting, the plant cell loses water either by evaporation, but not by osmosis. So take note of the difference. For instance, if the plant cells, if a plant is placed in a very hot place with less water in the soils, the leaves will lose water to the atmosphere by transpiration, or you can say by evaporation. Now, when they lose their water by evaporation, then the cells will collapse, and even the leaf will tend to droop or to fold. Then we call that one wilting. So wilting occurs when a plant cell loses water by evaporation, so that the whole of the cell wall collapses. The cell wall even collapses. Whereas plasmolysis occurs when the plant cell loses water by osmosis. The cell wall does not collapse under plasmolysis. Then what about linked genes? Now we have linked genes and sex-limited genes. Now linked genes, these are genes that are inherited together. They are not inherited independently. Linked genes, these are genes that can be inherited together. They are linked. For instance, someone can be tall, or maybe you can go in a population, you find all tall people are dark-skinned and all short people are brown. You try to move around, you don't find anyone who is mixing. You find if you are tall, then you must be dark-skinned. If you are brown, then you must be short. So what does it mean? It means that the gene for height, the gene for tallness is linked with the gene for dark skin. That is what it means, that the gene for tallness is linked with the gene for dark skin. So it means when the person is inheriting height, this height goes together with the skin color of darkness. So that is what it means. Linked genes, you find that two different characters, the genes controlling two different characters are linked together and they always move as a whole. So those are the crosses that we always do like this. So you find something like this. We say this is A, then this is B. Then that is one allele. Then another allele, this is A, this is B. So you find that now this one, and this one, these are two different genes controlling different characters, but they are found on a single loci. Those are the linked genes. Then okay. Then what if I say, Jaffa is saying that what if I say wilting occurs when the plants lose water in the atmosphere by evaporation that it can absorb from the soil. No, that is water stress. That is water stress. Water stress is when a plant loses more water than it can absorb. Wilting is not for the plant. Wilting is for the cells. Then, what is the effect of carbon dioxide on nitrogen concentration in a plant? No, it doesn't have any effect. Okay. Now I think we can now go to our question, then we wrap up. Okay. But now I think now I want to try out this question in your free time. This one now is our food for thought. Let us go with this one. This is food for thought. So I'm going to share this question paper on the group, then we'll try it out. So otherwise, I think we can stop from here. We're supposed to end at 4. It is now 4:20. Okay, so thank you for participating. Otherwise, I wish you a nice day. So, bye-bye.
Bye-bye, sir.
Goodbye, sir.
Okay,
Bye, guys.
Excuse me, sir. Are we having another lecture tomorrow?
Thank you for attending biology.
Sarah,